sorghum breeder based at University of Queensland, Hermitage Research Station. He's going to talk on integration of, or I will say, interdisciplinary research to drive the crop improvement, basically sorghum, and using BCNAM as an approach. So, Jordan, David, to you. Thank you.
Okay, well, thank you for the... great. Thank you for the opportunity to talk to you about our research in Australia. Thank you. So the structure of my presentation will be initially to talk about, I guess, the framework that we use in Australia to structure our plant improvement, crop improvement, and then, as I'm part of a genotype by sequencing workshop, talk about a potential application of that technology. So, for those of you who don't know Australia well, it's an extraordinarily variable environment to grow crops in. The program that I lead targets an environment that varies from greater than 10 tonnes per hectare to less than 2, and often we're trying to do that with the same 1 or 2 varieties. So that's very, very challenging, and there's some of those environments can occur at the same locations. It's difficult. So the sort of framework that's evolved to deal with this challenge has been this idea of G by E by M, and you will see numerous papers by people like Graham Hammer talking about this concept. And basically what we're trying to do as crop improvers in Australia and really all over the world is to identify favourable combinations of varieties or genotypes and management practices in a complex system where the resources available to search for those good combinations are limited. So the things that drive the complexity. First is environmental variation. So the type of environment experienced by crop varies according to the location and from season to season. And there's very little that we can do about this. Management systems, so agronomic interventions like fertilisers, planting dates, spacing, populations. The value of a particular management system will depend on the genotypes that are grown in it and the patterns of environments experienced by them. But we can change the management system. So we can change management systems, we can change genotypes. Genotypes. So as I said, the value of a genotype, we can change genotypes, but the value of a particular genotype will depend on the environment that it's grown in and the management system that it's grown in. But again, that's something that we can influence. We can change different genotypes and pick the best. If we go down a level of scale to gene networks, genes don't act in isolation. Different combinations of genes can produce the same phenotype. Interaction between genes occurs at different levels, and scales within networks, and between what we call subtraits. So, root architecture interacting with flowering time to influence yield. So the value of a particular gene will also depend on the environments and management systems and genotypes that it's deployed in. We can modify these networks and select the best if we know what the best is. So the constraint is you've got all of the permutations and combinations of all of those factors happening, and our capacity to search through those combinations is limited. So agronomists tend to evaluate different types of management using a limited number of varieties. Plant breeders evaluate large numbers of varieties but limited number of management systems. Genomic researchers and trait biologists are only able to look at a small fraction of the genetic variation and combinations that are available to them. And often they have to work in systems that have limited linkage to real genotypes and environments. So there's this disconnect between the development of management systems and the development of genotypes. So and the result is that changes in management systems can compel changes in varieties. So in Australia, we moved to this system of skip row sorghum, which is wide row sorghum, to make use of water in the interrow. And what we found is many of the varieties that were adapted to solid planting were no longer adapted to this new management system. So we had to change varieties. Similarly, there's a disconnect between the development of genotypes and management systems. So when we developed midge-resistant sorghum. That allows us to have different planting dates, and some of the planting dates were much more favourable. And so changing midge resistance gave us the opportunity to change management systems. But they were disconnected. They happened in sequence. There's a disconnect between the detailed trait biology work that physiologists do and performance in the real world. So here's some root chamber experiments on root angle, and changes in that trait may be positive or negative depending on the genetic context that trait's deployed in. So this idea of context dependency is really the thing I'm trying to get through here, that there are a large number of combinations and permutations, and we're trying to find the best one. And it turns out that the interactions are really a very important part of the system. Although there are main effects of each of those things, the interactions are important. So this is a great example. Some work published out of Pioneer Hybrid by Duvik et al. Looking at yield improvement in US maize. So there are two different management systems. The triangles are the current management system, high intensity, high density, fertility. The round closed circles are the agronomy of the 1930s, and the points represent hybrids that were the king at those particular years. So you can think of the red line as a baseline. This is the effect of taking a 1930s variety and giving it 2000 agronomy. This is the effect of taking a 2000 genotype and growing in 1930s agronomy. And that's the impact of the combination of the two things. And one of the reasons, and this is my feeling, is that this sort of effect is happening across a range of our production environments, but in most of our cases, we don't have this huge environment to work with as they do in the US Corn Belt. So they can select for the combination. They have been able to select for the combination over time. So there's a real opportunity to make progress by looking at the interaction. So how do we change the game? Because we don't have the money that Pioneer Hybrid has to select varieties, and by brute force. So we have to start thinking about smart ways to do it. And we would propose that using an integrated approach with linking technologies that build bridges across scales and disciplines. And really the fundamental parts of that we're working on are crop modelling and genomic resources. So if we talk about crop modelling, and we use the AppSim platform, it gives the capacity for us to understand the environments the way the plants see them, to predict the performance of specific genotypes in combinations of environments and management systems in silico, to explore those combinations and pick the best bets to actually do field research on. So it's more of a filter rather than reducing the number of combinations you want to have a look at. And also to dissect complex traits, for example, yield or various parts of water capture, into simpler traits that are more amenable to both breeding and also selection. So, for example, we could have some trait biology around root angle. We can look at changing the crop model, which is essentially growing a virtual plant, and use that variation in trait biology with the best knowledge we have of the other parts of how a crop grows, and management options, and historical weather data in our case, so we can get the variation that we know happens in our environments. And we can start to ask these sorts of questions. So what is the type of architecture that would work best in my current environmental management system? What combinations of root angle and row spacing would give the best yields on average in a particular location? And what happens if I plant earlier? Those sort of questions. What happens if we change the phenology? What are the likely risks? Because risk is very important to farmers in Australia. Risk is very important to farmers who rely on their harvest to survive. So you can actually get a handle on risk. So genome resources and high throughput mapping, comparative biology also allow us to link between experiments. So the same allele that may have been expressed in a experiment to look at root angle is expressed in a crop plant being grown in a field trial. So we can see which allele is present in which genotype in which field trial. We can look at the frequency of those alleles in our breeding populations. We can link trait biology experiments to real-world breeding programs and dissect complex traits into smaller components. Okay, so this is basically what I've said. You can start to ask these questions. Are the genes for the root architecture, for example, segregating in my breeding program? Am I selecting for a particular root architecture? So do a retrospective analysis to see, 10, 20 years of selection, are we going narrow or wider? Are there sources of a particular trait that I should look for? Does the variation in these genes have different effects in an early flowering variety compared to a late flowering genotype? So there's a whole range of things that allow us to really join data sets together and get a lot more power than we currently do from experiments, particularly breeding experiments, which tend to only get used once, and then the data is put on the shelf. Okay, so that was a little bit of philosophy, maybe too much, but I think it's important. I was quickly going to say some things about the core breeding program that I work on, which is a germplasm enhancement program now, but it's got a long history. So approximately 70 years of breeding activity, and the emphasis has changed from producing cultivars, because we live in an environment where we've got commercial companies coming in, so we don't want to compete with them. We want to be doing things that are complementary. Initially, before the companies came in, we were doing cultivar development, then it moved to hybrid parent development, and now it's a combination of germplasm enhancement, parent development, and integrated trait discovery. The traits that we're primarily interested in are, well, midge resistance has been important, but also the drought adaptation and grain yield. And secondary traits are around grain quality, ergot resistance, and a little bit in forage. It's been a very successful program, particularly under my predecessor, Bob Hensel. 100% of Australian sorghum hybrids have some genetics from our program. 80% of the hybrid seed sold in Australia currently has one parent from our program. We licensed 1,300 lines to commercial industry. Germplasm from the program is being grown all over the world in commercial hybrids, but also being used in breeding. So on the trait discovery side of things, we put together this integrated set of genetic resources, including mutant populations, our core breeding populations, a diversity panel, some sequenced genomes recently, conventional mapping populations, and a NAM population. So I'm just going to talk about the NAM population now. And again, give you a little bit of history and motivation for how we came to build this NAM thing. So when I was doing my PhD in the mid-90s, I was interested in using molecular markers, actually RFLP markers for those who are old enough. And I was interested in genetic diversity. So we looked at diversity in elite Australian sorghum compared to what was available internationally. So the blue lines are grain sorghum R lines, and it should have been pink, but it's red, are the grain sorghum B lines, the female lines. So you've got hybrids, so you want to have those two heterotic pools separated. And so that was great. Those lines are, I guess, represent US germplasm of the 1970s, 60s. But when we looked at what we had done to our breeding program, we'd really, by selecting for midge resistance and stay green, we'd ended up with a very narrow genetic base. And that potentially reduced our capacity to make gains. And yeah, as I said, the reason for that was selection for midge resistance, selection for stay green, and other agronomic traits. And also the small effective population size in breeding populations. Ooh, a little bit of a problem there. These are just some graphs showing the relationship between reduced diversity and increasing midge resistance, and the degree of polymorphism between the two parents of hybrids and increasing midge resistance. So the hybrids were getting more similar to each other, and the parents of the hybrids were getting more similar to each other. So we wanted to increase genetic diversity of elite germplasm while retaining adaptive traits, the midge resistance and the staggering, which are quantitative traits, and so they take a long time to stack up, and you just don't want to smash those things up. So how are we going to do that? As I said, this issue of context dependency comes up. There's no absolute relative merit value for a gene or trait. It depends on those things. The value of a gene depends on the context. So genotype, environment management. Context dependencies occur at all scales, and they're really the things that generate the major challenges for plant breeders or crop improvement programs. So epistasis, heterosis, G by E by M. So here's a good example of context dependency. This is some simulation data, but it's based on pretty strong biology, and this is using the environment information from a real breeding trial. And in fact, I could overlay the points, and they sort of fit in this area. And what we have along the bottom are different maturity groups, so 45-day maturity, 48-day maturity, 51-day maturity, across to 60. And each of these categories represents a level of tillering, so from 0 tillers to up to 1.5 tillers. And what we see is that at early maturity, tillering is favourable. So if you have a gene for tillering and your plant's early maturing, then tillering is favourable. If you've got late maturity, so this is a terminal stress environment, and what happens is the tillering plants use more water, they run out of water and crash. So a late maturing variety is in the field for longer, it's using more water, and so when a high tillering gene is deployed in that genotype, it's actually negative. And you can see there's a real potential inefficiency for plant breeding programs in that, do you select for high tillering? Do you select for low tillering? It's an inefficiency, but it's also a possibility to be exploited. I mentioned heterotic pools. So we have two different heterotic pools in a hybrid crop like sorghum. And whatever you do with diversity, you need to consider that because you want to make sure that the heterotic pools stay separated. So again, that's a different type of context dependency, genetic context. Here's a more complex diagram. This is showing different trials. Each box represents a different trial and different male parents. So this is an environment across the top. That's a male tester. So we're talking about hybrid combination. And in this case, in this single environment, and we're looking at the relationship between staggering and grain yield. So in this same environment, we have a significant negative association between staggering and yield and a significant positive association between staggering and yield simply by changing the male tester. Now we look at the same male tester, but in different environments. So again, we get significant positive association between this trait and yield and significant negative association between this trait and yield. So what we find in Australia is generally the positive associations between grain yield and staggering outweigh all the rest of them. But without, I guess, the benefit of 20, 30 years of breeding experience, you wouldn't be able to see that. Whereas this is eminently can be modelled. So back to our problem of trying to diversify. So we were trying to diversify, but we needed to increase the genetic diversity using exotic lines which were unadapted to our environments. So we wanted to do that, but for the breeders in the audience, as soon as you make a cross like that, you lose a lot of adaptation, and particularly those complexes that we built up over many years, staggering, midge resistance, but key agronomic traits as well, like flowering time. And how do we keep those things together, but also bring in diversity? And we wanted to be able also evaluate new genes from the exotics in genetic contexts that was relevant to our environments and management systems and populations. So we really wanted to see exotic genes in our elite background. And we also, because our interest is in actually getting things out there to farmers, we wanted to have material that could be used in breeding relatively quickly. And finally, we also wanted to, this is at the time when molecular markers were coming around, and we wanted populations where we could see a potential to use marker technology in the future to contribute both to breeding but also to biological understanding. So I'll talk about the resource that we decided to build. So again, at the time I was doing my PhD, Tanksley put out this nice paper on the advanced backcross method, and I got really excited about that. And we, after my PhD, we thought we would actually try and do that. And the method we used was to produce a very large backcross population, and we found, and with a single wild relative. And what we found was we didn't need to do more than one backcross in sorghum. A single backcross with selection was sufficient to actually get really good material. And so we thought, well, instead of doing one population of 300 lines, let's do 300 populations of 50 lines. And that's what we did. So we used this method where we took our adapted parent, we crossed it to an unadapted parent, we produced an F1, and then backcrossed that F1 to a genetic male sterile version of our elite parent. So we were able to produce huge BC1 F1 populations. And then we selected those populations for the key things that allow us to measure yield in our environments, which is basically maturity, height, and lack of shattering. but very little selection other than that. So we produced populations between 50 to 100 individuals, and we evaluated those in hybrid combination in multi-environment trials. So each year we would do approximately 20 populations, and we would evaluate those in trials. So this is a picture of what the structure of those populations would look like. So an elite parent crossed by a number of exotics, producing different subpopulations, which are mosaics of elite parent and exotic genome. But because of the backcrossing, it was on average 75% or better elite parent. So we're capturing lots of that good elite genetics that we want. So capturing as much of the those traits that we built up over years. So why backcross? And that's what I just said there. On average, it'd be 75%. Reality was 80%. And we can recover enough lines with the appropriate maturity. If we'd done an F2, we would struggle to find lines that would be acceptable for us. It also we found later, it simplifies the genetics so that that really helped actually in mapping genes because you get rid of the exotic by exotic epistasis, and you're really only looking at allele value within the elite context. And from the point of view of further trait dissection, it was pretty easy to take this material and develop isolines. Each of these box plots represents the genome of a different subpopulation, and you can see on average we ended up with 80% elite parent, 20% exotic. And this is the most extreme example. This is a population that we started originally with when I got excited about advanced backcross with wild relatives. Sorghum arundinacea is basically, or verticilliflorum it should be really, is basically a weed. That's what the F1 looks like. And that's basically what the F2 looks like too. Here's a selected backcross progeny out of that contains, at least 10 to 15, 20% exotic. So that was pretty amazing. So how did we select the exotics? We used a range of strategies. We used visual phenotypic diversity. We used phenotypic extremes from published or unpublished studies in physiology, grain size, various traits. We used elite lines from other breeding programs in other countries, including material from ICRISAT. We used geographic diversity, racial diversity, and also a small, a proportion of fertile wild species. And these black dots represent it's data from the CIRAD core collection, so diversity plot of sorghum diversity, basically. And the dots in red represent primarily, not entirely, but mostly lines that we have used in our backcross populations. So we've really done a pretty good job at sampling the diversity of sorghum worldwide. And recently we've had a project that has resequenced 36 of those parents. So this is part of an ongoing breeding activity that the primary target initially was actually to produce material that could be useful in a breeding program. As I said, we evaluate 20 populations in multi-environment trials each year for a range of traits, and we did it in such a way that we evaluated 15 new populations, 15 new populations and 5 populations from previous years. So that allowed us to analyse these trials as a single mega trial, I guess. And in later years, we actually conducted environment characterisation so that we were able to run simulation models at each of those sites to further add to the value of that data. Again, this is one of these box plots. Along the bottom, we've got height in centimetres. Each one of these boxes here represents an individual population from this program. The black line down the middle is the elite parent hybrid. So that's the height of the elite parent hybrid. This is the variation of the populations, and you can see that we've squeezed up the variation pretty well to within 20 centimetres, and mostly it centres around the recurrent parent. Similarly for maturity, but in this case our elite recurrent parent was probably slightly later than we wanted, and we've actually pushed the maturity earlier. But you can see it's still relatively narrow maturity range. Then if you look at yield, and this is data from a paper that we put in Crop Science, but these are environments classified by relative performance of genotypes. So environments that rank genotypes in a similar way were clustered together, and they really represent a high yielding environment, which is environment 1, something like this, and environment 4 is more of a low yielding environment. No, got it the wrong way around. Environment 1 is a low yielding dry environment, and that's where our elite parent is adapted, and environment 4 is more of a high yielding environment. So the interesting thing was that we found that even with our most wild crosses, for example, Purpurea cericium, which is a wild species, Drumundii, we were finding individuals within the population that were as good, if not better, for yield than our recurrent parent hybrid. Now, this wouldn't happen in all species, but in sorghum, it does. So that was pretty encouraging. So now, as the resource stands, it's around 4,000 lines and growing each year. More than 100 exotic parents have been evaluated. We've got some dart marker data on about 1,300 lines, and we're planning to GBS at least 1,000 in the coming year. We've got data from more than 50 field trials with greater than 2 million phenotypic data points. It's not quite up to genotypic data points, but there's a lot more pain in generating phenotypic data points. Some of those trials have environment characterization, and the reference parent and the female tester and 26 exotic donors have been resequenced. We've also licensed 500 lines from this stuff to commercial industry, so that's pretty good. So a little bit on, for those who don't understand the concept of NAM, a bit of background. So conventional QTL analysis uses recent recombination between 2 different parent lines to identify QTLs associated with a trait. And it has a range, and there are plenty of reviews. You can see this has high levels of LD, which means that you don't need a lot of markers to get genome coverage, and you can find QTLs relatively easy because of the replication of the alleles. They basically have 2 alleles at high frequency. But it doesn't have high level of resolution, so your ability to fine map is limited by the number of recombination events. And the other disadvantage of the conventional real population is that you're limited to the alleles that are segregating in your parents, or the parents of the population. And you only get to see 2 alleles. So association mapping is very popular recently and used in human genetics as well. Uses historical recombination, and it's performed by scanning the genome for SNPs, so you really need a lot of markers to do this well, that are in linkage to equilibrium with the trait. And it means that you have a lot more precision, so you can get close to the gene, but the power is lower and the potential for false associations is high. And there's a bit of wrong text after down there, but anyway. And also the power to detect alleles that are rare is also relatively low. Okay. And probably most importantly for a crop like sorghum, for many traits, the genetic background effects, this context, really limits your capacity to phenotype. So if you've got things that differ by, in our context, even a few days in flowering time can drastically influence yield performance. If you're trying to do yield on things that vary by month, it's a waste of time. So at the Buckler Lab, Jamin Yu has a nice paper on this idea of nested association mapping, which combines linkage analysis with a high resolution association mapping. And there are a range of different genetic designs that you can do. Many are what you call a reference design, where there's a single parent that everything's crossed to. And there are various subsets of different allele, diallele designs, so chain crosses, nested designs, all sorts of things that would be possible. But it turns out that the design that we developed for our what we call our diversified gene pool is really our form of NAM, and I guess you could call it Backcross NAM. If you want to do the association part of nested association mapping, you need high density marker data or resequencing, and so that was one of the reasons that we went into this resequencing partnership with University of Queensland, DAF, which is the organisation that Emma works for, and that I used to work for, GRDC, which is a Grains Research and Development Corporation, so it's funded by Australian growers, and Beijing Genomics Institute. So we sequenced 44 genotypes, including 26 of the NAM parents, and got quite deep sequence coverage. And so potentially we can do the association part in the future. So what's the opportunity in using exotic material? So this is data from that resequencing project where we split the genotypes into two groups, so improved inbreds, wild and weedy, and land races. And if you look at this, 59% of the SNPs, so diversity potentially, in our sample were unique to the land races and wild and weedies. So we're really only playing with a relatively small proportion when we work with improved inbreds. So while you don't want too much diversity, there's certainly a lot of material to exploit if you've got a way of exploiting it. So here's an example of actually doing some trait dissection on a quantitative trait with the NAM. And just to refresh your memories, it's basically like multiple small mapping populations. And flowering time, it's trait controlled by multiple QTLs. And when you do a real study in sorghum, and there are a number of them out there, people find between 3 to 5 QTLs for flowering time. But what we know from Arabidopsis is it's a complex trait, and we would expect to see many more QTLs. This is just an example of one of our mapping studies, so 3 QTLs for flowering time, and that was like 300 individuals, 2 environments. So again, if you remember back to my description of this NAM population, we actually did selection for flowering time. So we reduced the range of flowering time down to a relatively small band, and we got rid of large effect genes like photoperiod genes. So we were left primarily with temperature sensitivity and possibly a few minor photoperiod genes. We had phenotypic data from 20 trials, 10 locations, and 24 BCNAM population families, so about 1,300 individuals. And when we did the QTL analysis, we found about 3 to 5 alleles at each. Well, we found 40 QTLs, but 3 to 5 alleles at each locus. The allele effects were relatively small, but still, that may well be part because of our selection. But still, it's interesting to know that there are a lot of relatively small effect QTLs. And the most interesting thing to us from this study was the really strong consistency with the QTLs that were identified by Ed Buckler and Maize. So here's our QTLs that we found. And this is an example of this particular QTL where the red line represents the recurrent parent flowering time. So we had lines that deviated significantly from the recurrent parent in terms of being slightly later. And then we had potentially 2 alleles on the negative side. So that was pretty interesting in terms of understanding the diversity in what is a pretty critical trait for adaptation. So if we looked at these are individual families, and these are numbers of QTLs, what we tended to find was that there was this balance of positive and negative QTLs that gave a flowering time about the right level for a particular genotype. And it means that when you start intercrossing these things, you release variation in flowering time. And that's problematic in a breeding sense. So this is to show you that and this is work that has been accepted for publication now. Maize is like tetraploid sorghum. So wherever there's a maize flowering time QTL, or sorghum flowering time QTL, there's usually 2 maize QTLs. And this is, it's pretty remarkable. The red and blue dots are flowering time identified by the Buckler group in maize for male and female flowering. And these are the sorghum ones. And we sequence mapped the Buckler closest markers back to sorghum and position them near our QTLs. So that's one chromosome. Amazing thing is that they all look very similar to that. 65% of the markers identified for flowering time in maize were within 10 centimole lengths of the midpoint of one of the sorghum QTL. Many of them were less than 1 centimole. And the interesting thing was that looking at the 2 crops, there was some really nice insights. I think I might have chosen this example to try and sell sorghum to maize people, because here the maize guys have said, look, there's a single QTL for flowering time, but this data would suggest, well, actually that's an area of low recombination, and there are actually 4 flowering time QTLs there. But the reverse is true. There are areas of the genome in sorghum where you've got low recombination, and the same thing happens in reverse. So I think the fact that we identified such a large proportion of previously identified flowering time QTL in sorghum suggests that we're capturing a lot of diversity. A high degree of correspondence between the sorghum and maize, again, suggests that we've got a large proportion of the genes that are out there. And I think that tells you something about the power of this approach for what is admittedly a quite highly heritable trait, but we're talking about pretty fine scale genetic dissection here. And I think the interesting thing, one of the interesting things to me was this evidence from the Allelic series at most flowering time QTL. And also perhaps the thing that struck me as interesting was that that maize decided to keep both versions of the flowering time genes functional when it could have decided to. So there's obviously adaptive value in having both versions, and I think sorghum deals with this potentially by having more alleles. So where we're going to next? Well, BCNAM will form a major element of our future breeding and trait dissection work. We're going to use GBS to increase marker density and impute the sequence data from the parents of our NAM onto our progeny so that we can potentially get to gene level resolution for some traits. We're going to start looking at different ways to mine diversity. And a key part of this activity will be to integrate and extend the data that comes out of this sort of BCNAM trait dissection to our breeding populations by tracing markers, but also in terms of the value of alleles via this crop modelling. So to finish off, I'd say there's an opportunity to completely change the way we do plant breeding by, or I should probably say crop improvement, because it's not just breeding, it's also management, by using new technologies to enhance integration across disciplines. So obviously this is the work of lots and lots of people, and here are some of them. Emma's in the audience, so she's really been the lead on a lot of the marker work. Graham's and Eric around the modelling. Andrew Borrell around staggering trait. Vijay Singh is a PhD student who did a lot of the root architecture work. Alan Cruikshank's one of the other breeders there. I should have had Bob Hensel as well, because